Okay. And if you could please take a look on the chat, I pasted there some of the things that were on the forum, which was question/assignment. So please take a look at that. Today, we'll do a hands-on session to prepare and run your pipeline. We went over the same pipeline last time conceptually, and today we are going to try and do it together. So welcome back, everyone. And today our purpose is to find the full genome sequences, prepare FASTA files that are ready for analysis, selecting the appropriate genomic sequences, and running a full pipeline and starting to look at some of the outputs from that pipeline. So before we start, again, we have the organizational page where we can track how things are going in terms of all of the participants. There's not a lot of us, so I think it would be great for us to get to know each other a little bit more and kind of see how things are going for everyone. I know that in the context of the summer, there's a lot going on. So hopefully you are having some time to practice and work on some of the independent assignments. I think that will really contribute to your ability to get something out of this program that you will potentially use in the future. So the organizational page, let's go really quickly. On the organizational page, we have the list of participants here. You can go to the progress tab, and here we have some of you, and I see that some of you are starting to do some of the educational theoretical coursework that we have available, and also practice some of the analysis on the server. So I just wanted to make sure that it's starting to make sense how to use all these different resources. And hopefully, if you are facing any kind of troubles as you go through this, you could let us know ahead of time. The best way to talk about any kinds of issues or anything that you don't know where to find an answer for is to put it on the forum. So we have this forum tab. For example, we have session recordings. The session recordings are posted after each session. And so you can see the last meeting that we had is posted right here. And there's also going to be some information about files that we use, or any kind of discussion or kind of practical next steps for anyone that would like to do something in between the sessions. So for example, after session 1, I ask that you all complete your profile and update your full name. That full name is going to also appear in your certificate, so please don't use just your email address or any kind of, an acronym. So if you use dashes, points, or anything else like that, that's also going to appear on those. And then last time, we started talking about the Ebola virus. We looked at several of the publicly available data sets on the outbreak from 2014 to 2016, and even saw some new sequences from the 2018 outbreak. So the purpose of that discussion was to get you to look at NCBI virus and NCBI sequence read archive, and to try to think about a couple of these questions. So the best way, again, to post any topics that you would like discussed, any questions that you have, or technical issues, is to do it there. And again, just by looking at some of the activity here, I see that you're only getting started. And I hope that today's session, which is going to be a hands-on session, will help the rest of you try out some of the things that we're doing together in the session. And so I'll be pausing and waiting for you to let me know if you are not able to complete some of the steps that we're doing together. Okay, so as I mentioned, we post here the updates. Before we go on to the practical hands-on, let's do a quick review from what we discussed last time. So far, we've learned about next generation sequencing applications in metagenomics. So we talked about several different types of metagenomic and viral sequencing to study the microbiome, and the virome is a part of the microbiome. So several of those strategies include targeted sequencing, when sequencing is focused on a specific organism, looking at the total DNA or RNA that might include some viral sequences, because viruses replicate inside the host cells, and so we expect them to have some presence in that data as well. And amplicon or environmental sequencing, which means that some specific genes that are unique to a group of organisms, or that represent some kind of a variable sequence that we can infer diversity of genomes represented in a sample. For example, we talked about 16S ribosomal RNA. So those are some of the sequencing techniques that we reviewed. We then talked about how from that kind of data, we can analyze the data to understand the different types of viruses present in a sample. We can look at diversity of different samples. And so we talked about some alignment strategies for database alignments. We also talked a little bit about mapping on a genome. We then continued to talk about multiple sequence alignment. So last time in the session, what we did was we talked about phylogenetic analysis. And that analysis could be done on either FASTA files of assembled genomes, or it could be done with SRA files, which are sequence read archives. So we took one of the samples. In fact, we looked at a sample that has reads in it. So it's the FASTQ file. And we talked about how we can map those reads to understand what's inside, and potentially by looking at differences from the reference genome, identify whether this specific collection of reads represents something that is similar or not similar to the reference genome, and how we were looking for these variants in the reads that we have in the data. So those reads actually represent a number of responses that these different viruses have to different environmental conditions. And we can use those to evaluate, for example, differences between different hosts or differences between different locations. And so we applied that idea to the study of Ebola. In Ebola virus collection of data, we took 3 different groups of files, and those were from Liberia, Gabon, and United Kingdom. And then on NCBI virus, we were able to identify specific types of full genomic sequences in a FASTA format, and take them inside NCBI, first of all, to do exploratory data analysis. So in the exploratory data analysis phase, we looked at accumulation of mutations, of those differences from the reference genome. We looked at how they could be separated into groups, and therefore identify which samples are more closely related to each other or separated from a different group. So we saw, for example, that older samples from 1996 overall were different from this other group from Liberia and United Kingdom. And so then we also discussed how the output of such analysis includes alignment of sequences that then we can explore. Once we have aligned them, we can start looking at specific differences between the different types of files that we used, preparing a sample or a sample pipeline where we included some of those files, so Gabon, Liberia, and then Great Britain. So we can see how the separation into these different groups could be found in the phylogenetic tree. And we talked about some of these steps of taking the FASTA files, aligning the nucleotide sequencing, sequences, converting them to amino acid sequences, and then eventually building a phylogenetic tree. So I wonder if anyone had a chance to take a look at the assignment posted on the forum. The assignment was to look at the example of the pipeline. So we ran this pipeline, hope that most of you had a chance to run this pipeline. So let me ask you if you had a chance to try this pipeline at home or wherever you're accessing these lectures from. So if you have tried it, you can, and successfully ran it, you can say yes. Or if you had some trouble trying to replicate this, you can write that as well. I'd love to know. All right, so a couple of you, Trinika and Maya, were able to run it. Great. Anyone else? Did anyone else have a problem running this? Okay. Okay, so some of you were not able to. Okay, so hopefully today we'll try to do this together and also check if there were any issues trying to understand every step and finding the files and making sure that we can also think about other ways to apply the same analysis to other data sets that we can find online. So another type of data that was for FASTA files, right? Another type of data that you could have from next generation sequencing, in fact, that is typically coming out of a next generation sequencer, is actually a FASTQ file. The FASTQ file contains reads, and those reads are stacked up in different positions on the genome, and those actually help us understand the variation as it really happens in a sample. So when we have a collection of reads, we can analyze those reads for similarity, and if we find a position that has a consistent change from the reference genome, we can start looking for those variants. And those variants actually define the genomic difference or distance between these samples, right? What we see in the phylogenetic tree is a distance, and that distance is associated with the number of changes that each genome has. So whenever we have those types of changes, we're actually talking about a number of different types of mutations, so to say, or variants. We have substitutions. Those substitutions can be of high confidence. For example, we have here a high confidence substitution. It is consistent across many reads, and it is just one type of substitution from the reference genome. And then we have sometimes a low confidence substitution. The low confidence substitution is when we have a change, but we are unsure of what type of a change it is. And so that could contain an N symbol, which is we don't know. And then sometimes you might have a evidence of insertion or deletion. That evidence means that there are a number of reads that contain the sequence that will not have some sequence present in the reference. And so those would be typically substituted by dashes. And we'll take a look today at some of the examples that we can then identify how specifically were these different examples or samples different from the reference. Now, because we took 3 different groups, we are talking about 3 different consensus genomes. Essentially, we will have a consensus for each group of samples and how that is different from the reference genomes, from the reference genome. There's another type of a problem or of analysis that we can solve using this kind of study when we have SRA files. SRA files are generated directly from a biological sample, and that biological sample actually represents a collection of different genomes that are present in that biological sample. Because when a virus comes into the cell and starts replicating, not every single genome that it reproduces is exactly the same like the virus that entered into the cell. So when we take samples, biological samples, we typically have a number of different cells. And so we expect to see a variety of different genomes present. There's an analysis type called local haplotype reconstruction using SHORA. And that analysis method helps us reconstruct the genomic types by assembling those reads into full genomes and understanding what subtypes or what haplotypes of that same virus are present in the sample. So today, we'll also talk about some of these alternative pipelines. And these alternative pipelines can include, for example, analysis of SRA files, where we can find consensuses in each group. We can also look for different haplotypes and study variation between those haplotypes. For example, in the Ebola samples that we already, we first of all looked at FASTA files, but when we look at FASTU files, we'll be able to look at haplotypes and how diverse those haplotypes are compared between different groups. And when we're talking about consensus genomes, we're actually going to be looking at consensus per sample, meaning how can we generate a FASTA file if we have an SRA file with a bunch of short reads? How do we actually get that full sequence? Okay, so before we continue, let's talk about any questions if you had from last time. I know there was over the weekend, probably a lot of other things that you've thought about. So hopefully this refreshes your memory. If you have any questions related to those topics that we covered in our last session, I'd be happy to answer them now. And you can either type them in the chat, or if you want to unmute yourself and ask them, I'd be happy to hear those as well. Okay, so if there are no questions,
I don't have any questions. Oh,
yeah, go ahead.
I don't have any questions. I just wanted to note that this is my first time getting started, and I do have to like kind of play catch up with what's going on. But I did do some research with Dr. Kim of Grambling. I don't know if you remember us, but I was just saying that I needed. Huh?
Paul Kim?
Yeah, Paul Kim. But yeah, I was having some technical difficulties with emails that weren't coming through. So I just received a bunch of emails yesterday that is now allowing me to catch back up.
Okay, yeah, excellent. So unfortunately, there are a lot of emails going out about all of these different sessions, including the links to join the session. And also, please take a look at some of those links that I just pasted with the forum. So all of the previous recordings should be there. All right, so let's talk about this first assignment. So we talked about the Ebola virus on the NCBI virus database. I think we might have mentioned coronaviruses there as well, but obviously you can find any type of virus if you just search there. So I had a few questions, and hopefully you had a time to kind of look into that. Maybe not, but let's take a look. So what is the first available sequence for an assembled genome for Ebola virus from a human host? Was anyone able to look into that? And I'm just going to open the database as we look at this question. Okay, so if you remember what we did last time, we go to NCBI virus to answer this question. Right, this is the link to the NCBI virus. On NCBI virus, you can either search by sequence or search by virus. So you would click here, and then here you can actually look by virus name or taxonomy. So just simply entering Ebola and waiting a couple of seconds, you'll see Ebola virus come up. And now you can see that there's a total of 3,152. And in the question specifically, it's the first available sequence of an assembled genome for Ebola virus from a human host, right? So before we go anywhere else, we can go down here to host, type in human. Okay, and now we're down to 2,432. We can also look for only complete genomes. And that way we get to 445. And then here, if you scroll over, you can look at collection date. And so if you just go to the earliest one is from 1976. Okay, so that was just a question to help guide exploring this database. So hopefully you'll have a chance to look into this database a little bit more and see it for yourself, looking for viruses that you think you can analyze and trying to think of questions that are going to help you that can be answered with some of this type of data. Okay, so the other part of the question was, how many different hosts can you identify with a full genome for all types of coronaviruses? And yes, I'd be happy to slow down. Was there something specific that you wanted me to repeat that wasn't clear or too fast? Okay. Yeah, so there was a host that we selected, and then we also want to look at only the complete genomes. So nucleotide completeness means that we look only for complete genomic sequences, because some of these sequences are going to be partial. You can look at the length of the genome here, and you can see how the length of the Ebola virus should be around 18 or 19,000 nucleotides. Some of them, as this example, have only 1,400 nucleotides. So if you look at this sample, which is only partial, you can see that if you go over to the full accession number, you can see that this is a partial sequence, and it only contains this one nucleoprotein.
So what would a incomplete reading tell us? Like, why would that be important rather than a complete one? Like, why would we do a complete one? Incomplete one, I'm sorry.
Well, sometimes you're looking at differences between only specific proteins, or sometimes you know that a mutation that is important occurs only in a specific region. So instead of sequencing everything, you just do an amplified region, and that way you can maybe save some money, for example, right? You can just sequence a lot more samples and see how prevalent that mutation is.
Okay, thank you.
All right, so for example, when we look at these different genomes, if we want to understand when the first genome was actually submitted, right, we identified the human host, and then also we can go to nucleotide completeness. Okay, Trinika, does that make sense?
Yes, sir.
All right, great. So select this partial, select complete. And now for the complete ones, right? So how do we identify what was the collection date? We just sort them by collection date. And so here we have 1976. So you can even look at this accession number, click here, and identify what was it. So it was the Ebola virus isolate, Ebola virus from Homo sapiens from Sudan in 1976. And then you can also see the bio project that will tell you a little bit more information about that first effort. Okay, so that was the first part of the question, right? The second part of the question was how many different hosts can you identify with a full genome for all types of coronaviruses? So again, the whole program is going to talk about a number of different viruses as examples. And every time that we talk about a virus, we'll try to use a data set from that family of viruses or a specific strain or a species of a virus. So if we want to change some of these settings here, right, we can just delete the virus. So now we will have only complete nucleotide sequences from the human host. And you can see that the total number is 39,456. But instead, we want to go to virus and search for all coronaviruses. So you can just put in corona. And so the coronavirus family is called Coronaviridae. So we can click on them. Okay, and so now it's hard from this table to understand how many different hosts. So if we actually look for, yeah. So if we actually click on download, we can download the whole table. Okay, so if I'm going too fast, let me know. But here, right, instead of the actual sequences, we can download the table, for example, in CSV format. Click on next and download all records. So here it tells you some of the options of what type of information you would like to include there. We're interested in different hosts, so this is enough. You can just click on download. And it will be a CSV file, which is a table that you can, okay, yeah, so when you click on download, instead of nucleotide, you just select CSV format right here and do next, and then download all records. And then next, and you'll get that file. Okay, let's take a look at what's inside that file. Okay, so here you just essentially have the same table. And in the host, turns out that we only downloaded the human ones. Oh, because we have this selected. Sorry about that. So let's delete that and re-download our table. Okay, so I had a host filtered out for only human. So let's download this again. CSV format. Download all records, and I'll just click on download here. All right. So now we have, you notice that we, let's close this one. Okay, so here again, we can filter. And you see all of the different hosts that are present here, including some that do not have a host. Okay, so now, since no one really had a chance to look into this more, I suggest that we'll just keep it for the next time. If for the next time, right now you know how to find and download the data, if you could count how many different species are in this Excel spreadsheet or this database, and then put it on the forum as your response to this question. So the main question is, how many different species, or sorry, how many different hosts can you identify with a full genome for all types of coronaviruses? Okay, and this will give you a sense of how diverse coronaviruses are and how they are potentially, also when you look at these different species, think about what types of species are these, right? We know, we've heard a lot about bats and pangolins and humans, obviously, but let's see what other different types of hosts are there available in this database. Again, finally, we talked about this other type of sequencing data, which is not FASTA files, but FASTQ files. They contain sequencing reads. And so they're stored in what's called the sequence read archive. So right here, try to find a bioproject with sequence read archive samples on Ebola or SARS-1 samples from humans sequenced by Illumina. Again, to simplify the number of available different data sets, how do we search on bioproject? Okay, let's just do this together. And maybe before I do that, was that clear about the different hosts? Were you able to download that same table and see the different hosts that we have here?
Yes, sir.
All right, excellent. Thank you for the response there. All right, so we have the final step here, try to find a bioproject. So bioproject is actually a whole database on NCBI. So if we go to just NCBI website, which is this one. Sorry, that was private. Okay, so it's to everyone. So we just go to NCBI, and here you have all databases, or you can go between different databases. Now, these different databases contain different types of information. For example, we have the full genomes. Those full genomes are going to be in the nucleotide or the genome database. Some of them are going to be collections of samples included in different bioprojects. So in this case, we want to look for a bioproject, but it has to contain sequence read archive samples. And so SRA is another database right here that has sequencing reads. And we want Ebola or SARS-1 samples from human sequenced by Illumina. So let's just take one of those examples. Let's stick with Ebola for now. So if I click on, if I type in Ebola here, you'll see that there are a lot of samples here, 4,384, right? So a lot of them are actually different. You can see here RNA-seq of Macaca fascicularis liver. And if you kind of go through, you'll see that there are actually SRAs from, it seems like the same project, right? So all of them start with the same beginning, but then they have some variations here from different tissues. So which means that actually it's the same project. So how do we know that these SRAs are organized inside a project? Again, let's choose this next one, for example. Here it says 8 Illumina runs for human, right? Homo sapiens. So we are not interested in monkey samples here because we have bioproject with sequence read archive samples on Ebola or SARS-1 samples from human sequenced by Illumina. So as an example, we can take this one. And right here, it tells you about the study. So gene expression profiling of blood of Ebola virus disease survivors and healthy donors by next generation sequencing. And right here, you have a link to what's called the bioproject. Okay, so if we click here, that could be an example right there, right? So the question here is try to find, right, and find a bioproject. So if you were to find this bioproject, that would be the correct answer. And here you can see more about what they were trying to do. Clinical follow-up of Ebola virus disease survivors revealed a persistence of clinical symptoms and higher risk of mortality. Long-term analysis of the immune and inflammatory profiles of Ebola virus disease survivors are currently lacking. Here we evaluate gene expression profiles in 26 Guinean Ebola virus disease survivors and 33 healthy donors. We show a persistent increase of several biomarkers of inflammation, immune activation, and gut tissue damage in Ebola virus compared to healthy donors living in the same area. Gene expression profiling in blood revealed a significant enrichment in genes associated with antiviral responses in Ebola virus. Overall design, and then they tell you how many samples are in total. And here you can see how many individual files you will actually have within this study. Okay, so are there any questions about the bioproject?
No, sir.
All right, thank you. So if you are going to start planning your own pipeline, right? The goal of today's session is to go through the preparation. Okay, there is a question. How do I get to the bioproject? Let me show you. So I first found on SRA database, right? I searched for Ebola. I have these different results. I have the monkey results right here, and then I get to the human ones. And again, I can see that there's probably a collection of them. So I can click on one. And right at the top here, this PRJNA, this is typically going to appear right here, and this will take you to the bioproject. All right. Okay, so are there any other questions?
Can we, I see that the top of the screen changed from SRA to bioproject. So when we're looking for a bioproject initially, can we do it just to go straight to the bioproject, change it to bioproject and type in human?
You can, but here's the thing. If you just search on bioproject, you are going to find all kinds of data sets related to Ebola. So you will see here all kinds of different types of sequencing or data types. And because SRA is going to be specific to sequencing read archive, they're specifically going to be only a part of them are going to be available on SRA. So for example, some of them might be FASTA files of assembled genomes. Some of them, what we were interested to do is to find the full FASTQ files. Some of them might be published as just gene expression omnibus datasets, which means they are already processed and they show you gene expression profiles instead of the raw read archives that we have available on SRA.
Okay.
So yes, you can probably go through here as well, but then going into this specific project, and sometimes you might not be able to find the source data. So whenever we get to a point when you're able to think of your own project, depending on the question that you're trying to address, you want to find the appropriate type of data that you would like to use to answer that question.
Okay, thank you.
All right, great. All right, so now let's talk about, practically speaking, how can we prepare some of these pipelines? Let's skip these. Okay. So let's do a practice project. Last time we looked at Ebola viruses. We talked a little bit about the different coronaviruses. So let's try to understand in terms of preparing the data for analysis, right? In the analysis that we're going to do today, we're going to try to find specific areas of genomes that we find that are different between SARS-1, MERS, beta coronaviruses, and the novel coronavirus, SARS-CoV-2. So how do we actually find and collect all of that data? And how do we organize it so that we can have a balanced representation between these different types of viruses? So here we already have coronaviruses. We have the complete nucleotide selected. Here we can add human, just to also filter it for only the human samples. And here we see some of them are, for example, human coronavirus NL63, beta coronavirus 1, and others, right? So typically when you want to prepare your analysis, you want to have a balanced representation. For example, you don't want to have 20 examples of one thing and then only 3 of the other thing. The other consideration is that obviously you want the length of those to be as closely related to each other as possibly, as whatever you can find. And if you want to have a representative, for example, from the SARS-1 or the SARS-2, the novel coronavirus samples, you also want to organize them in groups that will not have any additional variation that you don't want to interfere with your analysis. So if your question is, how can I identify differences between these different types of viruses, you would want to be able to look at how, for example, 10 samples from China, 10 of novel coronavirus, 10 samples of SARS-1 also could be from China, right? So if you want to limit it to the variation within one country, that will eliminate some additional potential biases that could be introduced in the analysis. So let's try to do that. Our first goal is to, instead of 10, let's do 5. So let's organize them by species here. We'll take beta coronavirus as our first one. So if we want to find 5 different beta coronavirus 1 samples, we can look at which locations we have here. We have the US, Mexico, and here we have China. So since we know that, for example, we have most of these have been detected in China, let's try to keep it within one location. So if we want to find 5 different samples from beta coronavirus 1, again, when you look at the dates, you're looking at all 2015, 8/31, but you might want to look here also for the collection date. They might be very different. And as you can see, 2012, 2005, 2007, there's a huge difference, right? So we already saw with the Ebola virus analysis that we did before, that if we take samples, they typically tend to separate by time, by date. So here we want to, again, minimize that variation within the group. So when I want to select some representatives of the beta coronavirus 1, when I'm trying to compare differences between these different strains of coronavirus, which ones would you recommend that I select? From 2007, 2012, 2010, which ones do you recommend that I select for this comparison?
2007.
Okay, and why is that?
It looks, I could be wrong, but it looks to be the most abundant.
Okay, so first of all, yes, you're right. That's a good question. Which one is more abundant? But given other types of sequences that we will be looking for from other strains, right? When you think about trying to minimize variance just because of date, for example, we're going to select also the SARS, the novel coronavirus, right? For the novel coronavirus, which dates would that include? since it was detected in 2019, you're likely going to only find 2019 to 2020. So when we want to find additional comparisons for that, we probably want to take some of the latest ones, right? So maybe instead of taking the older ones, and maybe those are more abundant, we would want to find whatever the latest one might be. So if we look for beta coronavirus, and here instead of coronaviridae, we can type in beta coronavirus 1, and then sorted by collection date. So you can see that there's 2019 in the US. And here we have some of the latest ones from China, and they seem to be all from 2012. Since this one is an individual one from the 6th months, but we have here a few from the 5th month and a few from the 3rd and the 2nd, let's select these. So we're looking for 5 from each one. So we're just going to select these 3. And we can also select these 2. So one of the ways that I can generate the selected samples as a single FASTA file is to click on download.
Sorry, why did we select those?
Well, because we want to minimize variation based on any other phenotype except for the one that we are trying to measure the difference in. So the differences that we want to measure in right now is difference between the different strains. Strains are beta coronavirus 1, SARS-1, MERS, and the novel coronavirus SARS-CoV-2. So if we want to eliminate all of the other variants might be related to the location. So that's why we only chose China. They also could be related to date. For example, we saw in Ebola virus how older ones are very different from novel ones. And so if we try to minimize the range in years, we know that we need to take the latest ones. Does that make sense?
So but why not choose the first 2019 ones? The first 5 of the 2019 ones?
Because they're from USA and not from China.
Okay. I'm sorry.
Right. So, for example, we could have focused on USA, and maybe there are some MERS one from USA, I'm not sure. But as an example, let's just go with China for now. Okay, so we have selected results 5, and we can click on download and select some nucleotide sequence data FASTA format. And then you click on next and only download selected records. So let's download this. And here it's just going to ask for the definition, and we'll take a look at that in a second. So we can download. You should get a FASTA file, which you can open in any text editor. Okay, so what are the 5? The 5 that I selected were, they were from China. So you can see from 2015. So KF9, 23902, 23903, 23904, 23898, and 23906. Okay, did anyone have questions about how to generate this actual FASTA file?
Could you just walk through it with us?
What was that again?
Could you walk through it with us one more time?
Yeah, so we search for the type of virus that we're looking for, and we want to compare the different strains, beta coronavirus 1, SARS-1, MERS, and SARS-2, or the novel coronavirus SARS-2. So then we want to find only similar sequences in length. So we will only select the complete ones, all from the human host. And here we want to minimize variation between these different sequences. So we're actually going to find those that are all in the same location as much as possible. So we selected China. And then we looked for the ones that will be closest by date. We know that the novel coronavirus is going to be from 2019 and 2020. So we went with the latest ones that we have available for China. So if you scroll down from the very top, you'll start seeing some of the China ones. If you scroll to the collection date, that's what's important, not the time it was published, but when it was actually collected. And here you can see this is an outlier because it's in month 6. We need 5 of them, so we'll take from month 5, we have 3, and then from month 3, we have 2. So those are the 5 that I selected for the analysis.
Okay, got it. Thank you.
All right, so then we downloaded this file, and this is a FASTA file. So again, let's take a look at what's inside this FASTA file. Every time there's an accession number included in a FASTA file, it starts with this symbol. So if you want to know how many of these you have, because by scrolling down, it's sort of difficult to find them. So you can just search, okay? You can do Command F, or you can go to search and replace or find. And here, just put in this symbol. And when I put it in, it tells me how many of these I have. And so I have 5 of them, and I can navigate between them and find all the ones that I selected. Now we can see how this process of navigating this specific database could be tedious, because we want to find 5 of each one, and we want to then go and switch it from beta coronavirus 1 to SARS-1, right? So it's going to take a lot of steps for us to get there. What is a better way, or an easier way to actually do this? So let's, instead of beta coronaviruses, go back to Coronaviridae. We know that all of the sequences are going to be within that range. We'll keep it human and we'll keep it complete. And let's do what we did before. Instead of just downloading specific ones from here, let's download the whole table. So as we download the table, okay, we already did this before. So we can just download all of the records. It's just going to be a table of accession numbers. Okay, we do want to know the accession number. We're not so interested in the release date. We're not so interested in species because we already, we're not so interested in host. Collection date is important to us. Geolocation is important to us. And length is going to be important just to check. So for now, let's just keep it like this. Okay, is everyone with me here?
Yes.
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 All right, excellent. So let's download. Yeah, go ahead.
Did I miss where we clicked complete, or we didn't click it on this one? Like the complete read?
Yeah, it's already filtered out for only complete ones, so we don't have to worry about selecting it again there.
Okay, I didn't see it, I'm sorry.
All right, so here we're gonna, instead of nucleotide, select CSV format. We just want the table, and download all of these records. And this just tells you the names of columns that are going to be included in that table. So we're going to click on download. And it's going to be, again, one of those tables, but it will have a whole bunch of different accessions in it. So now let's take a look at how we can use this table to actually select the files that we want quickly and download all of them in FASTA files. So here you have that table. You can see that we can filter by geolocation and by species. So here's SARS, which will be SARS-1. If we scroll down, well, actually, let's just create a filter. Okay, let's see what's inside this column. So we have the beta coronaviruses, MERS, SARS, and other types of coronaviruses right here. So if we want to select only beta coronavirus 1, right, here we can just select beta coronavirus 1. Here only select China. And so here are all of the different ones that are available. You can see how the dates are going to be a little bit different here, but what we want is the latest ones. So what are the latest ones? They seem to already be the latest ones here on top. So all we really need to select the first 5, or we can skip that first one, 1, 2, 3, 4, 5. We just take these accession numbers and place them in a simple TXT file. Okay, so once we have this file, it should be just a plain TXT file, right? And we can save it. For example, I'll call it Beta China 2012, okay, or Beta Coronavirus. Now, after I prepare the ones for beta coronavirus and all the other substrains, and I have individual TXT files like this, or even one single file for all the ones that I would like to select, I can go and fetch those on NCBI. So on NCBI, there's a specific site dedicated to batch retrieval of files. This is the link. I'm just going to paste it right here in chat. Okay, so you can find here on top which database you want to download the samples from. You upload a file, just like what we created now with those accession numbers, and then we'll retrieve them. So let me show you how to do that. Okay, I saved it actually on, yeah, right here. So I just take that one file and I click on retrieve. And you can see that it received 5, passed to Intrez 5. It means that it didn't find any missing ones. And then I can just retrieve records for 5 UIDs. We're doing 5 now just as an example. Obviously you could do, 50 or 100, right? It really makes it easier to download quantities of these different files. And now once you get a list of them, you can either go inside and download them individually, or you can click on Send to, File, and then Format. You can select FASTA, Create File, and it will download to your computer. You'll essentially get the exact same thing, right? So these are two different ways of doing the exact same thing. One is based for exploration, when you're exploring it on a database and you want to validate everything right then and there. But once you kind of know what you're looking for and you have many of them to deal with, it's much easier to download the table, select the ones that you want, organize them into a single file, right? Instead of 5, again, you could have 25 here and download all of them at the same time. Okay, so I'll now show you how we can take such a FASTA file and actually build a pipeline. But before I do that, are there any questions about what we just did?
No.
All right. Okay, so if there are no questions, let me just show you already a prepared file with all of these sequences. Okay, so here I have a file. You can see that I already selected a bunch of accession numbers, but it's not 5, it's more than that. All of them are organized according to MERS, SARS, COVID-19, and beta coronavirus 1. Okay, I'm going to go to the same link to batch entres, and I'm going to upload this file. So this was okay, so here's the file. Choose this file, retrieve. So it has 22 sequences in there. I'm going to retrieve records for 22. You can see that there are different types. Some of them are from the Middle East, other ones from other places. And then I can go to send to, file, and the format would be FASTA, and create the file. Okay, so now I have a downloaded file for all the sequences that I would like to compare. What else do I need to be able to perform my multiple sequence alignment? One other thing that is useful to have is a reference genome. The reference genome could come in a couple of different formats. It could be a FASTA file, just containing the nucleotide sequences, or it can contain all of the different elements that are typically available in an assembly file. So this is an example of a GenBank file. And here you can see a lot more information that we saw on NCBI as well. Here the sequence is in actual amino acids and not in nucleotides. So if you scroll down, you'll see how all of this sequence is also organized by gene. And at the very bottom, you have the nucleotide sequence. So this is going to be an important file for me to have for analysis. So how do we actually find the novel coronavirus sequence, reference sequence? So right here, you'll see a resource page dedicated to SARS-CoV-2. And you can see that there's an NCBI reference sequence for SARS-CoV-2 genome, and that is its accession number. Okay, so here you can view the sequence record. And again, depends on how you would like to analyze your data. You can use SARS-1 as your reference, or you can use SARS-2 as your reference, or you can use other ones as well. But here we have the full genome, And as we mentioned, it's useful to have it in a GenBank format instead of a FASTA format. So here you can click on File, and the format would be GenBank. You can create a file. Obviously, saving all of these files in a way that you can then find them easily on your computer is also important. So instead of having a bunch of different sequences and looking in them, right, it's good to have names. So once we do this step of preparing the data for analysis, and we know that we've selected an equal number of sequences, the sequences are complete and they're high quality, and we would like to understand how they relate to each other, which ones are more similar, which ones are more distant from each other. Let's go to start the analysis. Okay, so we already used this last time, so all of you are probably familiar with analysis stages. And here we have two different sections dedicated to phylogenetic analysis. Phylogeny is not just for viral sequences, so you can use it for distances between genes in different species, for example. One interesting analysis in the context of SARS-CoV-2 might be comparison between ACE2 receptor sequences in different species. Or we can look at distances between specific viral proteins. So we don't have full genomes, we only take a specific protein that we're interested in. For example, the spike glycoprotein, important for cell entry, that might be interesting to compare. But in our case, we want to work with full genomic sequences. So we can either do it here under phylogenetic tree. Okay, and last time, I believe we prepared a pipeline like that. So now let's take a look at how do we take the sequences that we just downloaded and do such a comparison with what we have now. So the first thing that we have to do is to change what type of data format this is. So this is FASTA file, right? And we need to then click on 2. And is everyone here, by the way? Is everyone still with me?
Yes.
Yes, sir.
Yes, sir. Yes.
All right, great. So again, let me know if I need to slow down. But here we have, first step, we select what type of next generation sequencing data. We need to upload. What would we upload here are the FASTA files that we just prepared. Okay, so those FASTA files would be on your computer somewhere. Okay, for example, mine are right here. So after we have all of our FASTA sequences, we only prepared one single file. So obviously we cannot separate it into different groups, but you also have the option to prepare them as 4 different groups, for example. And if you have 4 different groups, then you can organize them by different groups and maybe do additional type of analysis. In our case, we're going to skip this. So we're just going to put in group A. As you know, we're not just going to do multiple sequence alignment of the genomic sequences, we're also going to use the conversion to amino acids. And amino acids are encoded in codons. So to do that, we can just use the GenBank file that we just created. And that GenBank file is going to contain the conversion between nucleotide sequence and amino acid sequence organized by different genes. Sometimes you will have samples that don't have good annotation in terms of collection date. So you might have, for example, just the year, or you might not even have the actual collection date. You might have a date of publishing of that sequence. So in that case, whenever you do this multiple sequence alignment and build a phylogenetic tree, it's very helpful to have some kind of a reference for the time point, right? So if you have a time point that will represent a date of collection, you can add it here as outgroup for evolutionary time approximations. And you want to have two of those, one for early time. For example, our earliest sample here might be from 2012, and from the late time, for example, from 2020. And so that difference between 2012 and 2020, 8 years, could then be used as a reference point for the mutational rate. Okay, sorry, we're gonna press on skip. We don't need that because we actually know the collection date. So now we can click on start, multiple sequence alignment. After we do multiple sequence alignment with nucleotide sequences, we can already build phylogenetic trees, but to have a better understanding of how those impact conversion to amino acids, we can select this additional step of multiple sequence alignment AA. We also have an option to consider only the third codon position. There are some publications referenced right here that tell us that there are specific selection, strong purifying selection happening at synonymous sites and specific to the third position. So this is more related to viral sequences if we understand how the type of virus we're using is related to the third codon position. So for now, we'll just keep it at false. Then we can also choose what type of a phylogenetic tree we would like to build. Each one of these methods is built on an assumption about a model that represents evolution, right? So this is an evolutionary study because it looks at common origins. So how one strain of virus might have resulted in several, and we want to trace the evolution between those nodes. And so here we have an ability to select between different processes. For example, we have a birth-death process. So the birth-death process is a process of progeny and death within a single population. So you can see that we have divergence over time, and we have a specific so here you can see both birth rate and death rate are present, and you have relationships between individuals from different species, which is kind of what we have. Birth-death process first estimates the lineage in time, the last common ancestor, and changes are measured in the directions of divergence. It is advised to use for speciation time estimation, so how time makes it more specific. And it is more sensitive than the Yule process since it has both parameters. So it includes both the parameter of time and the mutation rate. And here you can select what type of sequence you would like to rely on for this analysis, nucleotide or amino acid. So we can actually choose amino acid here, click save, and then you can click on end. An alternative to choosing a specific type, and by the way, if you want to go back one step, you just do right click on wherever you selected. You can include all of your phylogeny methods and then compare the results between those. If you select all phylogeny methods, because they are going to be based on specific types of models, you can also evaluate the different trees, and those different trees could be estimated to construct a single median tree between all the possible model predictions of trees. And that way you can have a better representation of different models because we actually are not thinking right now, when we are considering which model to use, which one is the most appropriate for the type of data that we're using. And then you can click on End. And you will also see how in many different publications, this type of a process is described. You don't just take take a collection of files, just do some kind of a preliminary analysis of a phylogenetic tree and assume that that represents the full truth. There are so many settings that you typically have in each step of the way that it's important to do that, select those settings with understanding, and then evaluate the different settings, comparing them one to another to make sure that the results of your analysis are, that you can be more confident in them, that they're reliable. Okay, so are there any questions about the pipeline? And were you able to create a pipeline of your own similar to what we're doing now?
Like I said, I was doing the research with Paul Kim, and so we got the chance to do the pipeline. And I got a chance to see how long pipelines actually take, so yeah.
Yeah, so the goal is to start it now, and you know, after you have it, okay, but were you able to start the pipeline? That's the main question. I don't want to, you know, I think as we move forward, not having a pipeline set up and started and completed will really limit your ability to work with some of the outputs that these pipelines will produce.
Your speaker broke up after I finished talking, so I'm not really sure what you said.
I'm just saying that it's important between now and the next time to be able to complete one of these pipelines and get the output, so that for the next sessions that we will do, we'll actually go into the different types of outputs that these pipelines have. And so, you know, I was just asking if everyone that's here on this session was able to actually start this pipeline. Okay, so Trinika is saying that she will watch the recording to finish the pipeline. Did anyone else create the pipeline?
That's what I'd like to say, Doc. I will go on and finish mine, but I have not finished it. But certainly, I think I need a little more of a review, because this is my first real exposure to this.
Okay, yeah, I understand. So that's why we wanted to do this again, so that everyone has a chance to practically start it on, you know, your own account, to then be able to track the output. So did anyone else start this pipeline? So, okay, apart from, so, okay, let me know if you were not able to do it. You know, I can repeat this again just to make sure that you do start it. Like I said, for next time, we're going to be reviewing the outputs, and the goal is to be able to actually reproduce this for your own data and see how the different steps play out in the results. So yeah, if you would like to review this again next time, please try and do this before we actually meet next time on Thursday. Okay, so again, you will have to see and wait a little bit for that outcome. It's not going to be, you know, just a few minutes and it's done, and it's going to go in a queue. So those of you that started first, you know, you'll get your results first, etc. So, and just remember that there are going to be some delays as you do this. Now, there's another section that essentially allows you to do the same thing, but is more specifically designed
for viral genomes and has a lot more capabilities for what we will do in future sessions. Okay, so I just briefly want to mention what those are. So here we have the phylogenetic tree, right? This is a universal section for any kind of a phylogenetic analysis. Here we also have virus quasi-species. And so the virus quasi-species is actually designed to do a little bit more. I just want to explain what are some additional things that you can do. So we saw already how we can take either FASTA files that contain full genomic sequences, or we can take FASTQ files that contain individual reads from the sequencing that have a lot of variation inside each sample. It's not just a single conserved sequence, it's actually multiple types of sequences combined in these different samples. So here, if we want to use that information inside those reads, we can actually calculate a number of different other parameters. For example, we can look for variants, like we saw in one of the slides previously, that allow us to study the mutability of an individual position. If that position is highly mutated or fairly conserved, gives us an idea of how it might play out in a population of viruses. We can look at comparison between groups of viruses to compare mutations that accumulate in these different samples. We can also look for deletions and long deletions, not just individual positions being deleted, but whole segments being deleted. They're called defective virus genome deletions. And then here you can essentially see some of the same methods that we dealt with today. We dealt with identification of haplotypes within, again, FASTQ files, consensus sequences from FASTQ files, and then also multiple sequence alignment of FASTA files to build this phylogeny that we just did. So if we think of the other types of sequences that we can use, I want to just give an example, right? Not based on those FASTA files. Sometimes FASTQ files could be fairly large. So I'm just going to paste a couple of links here. When we deal with FASTQ files that could be fairly large in size, so downloading them and uploading them to our computer might take a long time. But we can get additional information from these SRA files because they contain actual reads, and we can evaluate those for a variety of reasons. So let's try and use these files that are already uploaded in the sequence. We're gonna go back to Ebola virus like we did last time. And here we can select, instead of FASTA file, we can select FASTQ files. Okay, just gonna build another pipeline so you can also try that. Here we'll use single end reads. Then we can click on NGS files. And here, instead of uploading a file from the computer, we can actually use files from a link. And so what I gave you are 2 links to files already on the server. So I'm going to paste the first link in one box and the second link in the second box, okay? Again, we'll start it. Hopefully by next time we'll get the results, so we'll be able to review those results in our next session. After we've uploaded them, again, we'll ask us to assign them to groups. We don't really need to assign them into groups right now, so just drag them into a single group. This is going to be Ebola virus, so we can skip this for now. Okay, and then here, again, it's going to ask us for an outgroup for evolutionary time approximation. We can skip that one as well. We will need a GenBank or a FASTA file for the reference. So we can use, I'll paste this information, by the way, on the forum if someone wants to reproduce this on your own. Okay, so here, sorry. Okay, as you remember in our first session, we used the Ebola virus example, so we can choose from that folder that we already had. And you click on submit, now you have, again, you can build a pipeline. So here we can click on start. After that, we have an option to pre-process. And when we pre-process, we eliminate some of the technical sequences. Instead, we can click on Shora, and here we'll be able to identify haplotypes. And what we'll see in our next session is how diverse these two samples are in terms of the different haplotypes present in each file. Don't have to change any settings right here for now. After those haplotypes were identified, we want to understand how those haplotypes relate to each other by aligning them to each other. Again, we can convert those to amino acids and select codon position. Okay, again, we'll use all phylogeny methods based on amino acid sequences and click on end. Okay, so I'm just going to call this Ebola virus haplotype diversity. Okay, so that's another pipeline that you can build. We'll take a look at this example next time again. So what we'll try to do from session to session is really go through some examples. We'll talk in some sessions about the biology. In some sessions, we'll talk more about the specific types of analysis and analysis results. This session was dedicated to a hands-on on how to prepare and run your pipeline. So we reviewed how to find FASTA files, either on NCBI virus, or how to collect a whole series of files in just NCBI batch entres. So hopefully you found that useful. And we looked at how we can compare different viruses from their genomic sequence or from the raw read format, where we can identify a lot more information from each individual sample. And next time, what we'll do is we'll start talking about the results of that pipeline. Okay, so we did one pipeline for coronaviruses in terms of multiple sequence alignment of different types of coronaviruses. You can either replicate the exact same pipeline or change a little bit the types of files or sequences or settings of the pipeline. And then we'll also look at how haplotypes could be different between these two different Ebola virus samples, and how are they different in terms of diversity and what kinds of sequences we can find in each one of those samples. So are there any questions about what we just did?
No, sir.
All right, excellent. Well, thank you all for joining again today. Hopefully between now and next time, all of you will be able to start some of these pipelines. If you have any questions, post them on the forum. I'd be happy to help you out and address any of those. We track all of the activity, and we also provided you with some online resources that can help you understand some of the technical details behind the data preparation techniques, as well as some of the types of viruses that we're going to be studying. So please take a look at those, and I'll be looking forward to seeing you again next time. All right, thank you, and I'll see you again on Thursday.